If I take a cell, in this cell, this is nucleus, cell is resting state. Suppose this is resting state, the cell is in resting state. In this cell, I saw the nucleus. In the nucleus, if I ask you that how many chromosomes you can see, the cell in resting state, yes, how many chromosome? There are 23 pair of chromosomes. How many chromosomes are there? 23 pair of chromosome. If I give you a cell is resting state, not dividing, and I ask you, can you visualize the number of chromosomes by any technique? Please do reply. Can you see how many chromosomes are there? Yes, can you see? Yes or no? You can't see. When cell is not dividing, resting state, then these 23 pair of chromosome, the genetic material is present in the form of chromatin. You can't see chromosome, can't see, because the genetic material is present in the form of chromatin when we stimulate the cell. When cell divide, prophase, metaphase, prophase, metaphase. Yes, in metaphase you all know chromosome appear on equator. Yes, chromosome appear on equator. So metaphase in cell division, yes, prophase, metaphase, cell, that these chromosome appear on equator. But when cell is not dividing, at that time the genetic material is present in the form of chromatin. Chromatin may be of two type. One is euchromatin and second is heterochromatin. Euchromatin or heterochromatin. Heterochromatin is condensed. Condensed means darkly stained. Transcriptionally active. Inactive. Transcriptionally inactive. Transcriptionally inactive. Euchromatin is relaxed state. Light stained. Transcriptionally active. So we found that euchromatin, heterochromatin, these are means two types of chromatin present in our nucleus. So in our nucleus I found that this is heterochromatin, darkly stained. This is euchromatin, lightly stained. So you can understand, look at me. This is histone protein. This is having positive charge. Double stranded DNA, negative charge. Due to these opposite charge, DNA is tightly attached to histone. This is histone protein having positive charge. So if I ask you, histone is having positive charge, DNA is having negative charge. Due to opposite charge, DNA is tightly attached to histone. Okay, and tightly attached means heterochromatin. Tightly attached means heterochromatin. Now, if you want to synthesize messenger RNA from this DNA, suppose I ask you, you want to synthesize messenger RNA from DNA which is in heterochromatin, you know you can't synthesize. I told you, if you want to synthesize, two strands will be separated, one strand will act as a template, so it requires space. But when it is tightly attached, then DNA can't synthesize. I told you DNA two strands will separate. Okay, but if it is tightly attached, it can't separate, can't synthesize messenger RNA, means transcriptionally inactive. Transcriptionally inactive means can't synthesize RNA. But euchromatin is relaxed state, transcriptionally active. Understood? Now, what happened? What happened that this was nucleus. In this, genes were there. Out of 25,000 genes, so many genes are kept in inhibited state. You know, gene expressed, only those genes are expressed which are required by the cell. Suppose there are so many genes in our nucleus, only few genes are expressed which are required, other genes are kept in inhibited state. And how they are kept in inhibited state? We may keep them, one mechanism is that we may keep them in heterochromatin. In heterochromatin, genes are kept in inhibited state. Why we are keeping these genes in inhibited state? Why we are keeping these genes in inhibited state? We know that if adverse condition would come, we will activate the gene, will synthesize messenger RNA, will synthesize protein, and protein will tolerate that adverse condition. Look at the screen. Yes, I told you lacoperon, we have so many genes which are kept in inhibited state, just like lacoperon. When adverse condition would come, these genes would be activated, synthesize messenger RNA, synthesize protein, and cell survive. This was prokaryote. In prokaryote, we saw lacoperon, that in lacoperon so many genes are kept in inhibited state. The simple mechanism was repressor tetramer. But we are eukaryote, little bit more advanced. We have different mechanism. We have nucleus. In nucleus, so many genes are kept in inhibited state in the form of heterochromatin. When adverse condition would come, suppose this adverse condition came, this gene will be activated to synthesize messenger RNA. But when it is present in heterochromatin, can it synthesize messenger RNA? No, it can't synthesize. Now I want to synthesize messenger RNA from this gene which is present in heterochromatin. How can I do it? Yes, we know that this is histone protein having positive charge, DNA is having negative charge. Now what I will do, we will take acetate. I will take acetate CH3COO minus. Acetate is having negative charge. This acetate will go and will bind to histone. We will do acetylation of histone. Acetylation histone. When we will do acetylation of histone, positive charge of histone will lose and DNA will relax. Why DNA is attached to histone? Because histone is having positive charge, DNA is having negative charge. If you attach acetate to histone, positive charge of DNA will lose and DNA will relax. Positive charge of histone will lose, DNA will relax, and relaxed DNA is called euchromatin. Euchromatin is transcriptionally active. So suppose adverse condition came, toxic stimulus came, this gene is present in heterochromatin. This heterochromatin, acetylation of histone occur, this become euchromatin, synthesize messenger RNA, messenger RNA go to cytosol, it synthesize protein, and protein go and destroy this toxic stimulus. Understood till now? So this is the mechanism how we express few genes. Express means DNA to RNA, RNA to protein. So this is the way how we express the gene. Okay, so many genes are kept in heterochromatin, which are kept in inhibited state. When we require these genes are activated, just like acetylation of histone, and these genes synthesize messenger RNA. Messenger RNA go to cytosol, synthesize protein, protein tolerate that adverse condition. So question was asked, acetylation of histone. Yes, answer please. Now suppose this adverse condition gone. This adverse condition gone. Toxic adverse condition gone. Now you don't require this protein. Adverse condition gone. Now, yes, now I will do deacetylation of histone. Deacetylation of histone, yes, when I will do deacetylation of histone, again, yes, it will have positive charge and DNA will bind to the histone protein. Deacetylation of histone, yes, again, yes, positive charge, bind to DNA, heterochromatin. Heterochromatin. Clear? Very good. So histone acid, which is true about histone acetylation, answer is increase euchromatin formation. Acetylation of histone increase euchromatin formation, and deacetylation of histone increase heterochromatin. So we can convert euchromatin to heterochromatin to euchromatin according to our requirement of gene. Yes, we are modifying, yes, the expression of gene by acetylation and deacetylation. We are not doing mutation, we are doing something above the genetics. That is why this phenomena is called epigenetics. So epigenetics is reversible, inheritable, heritable, modification of DNA or histone without mutation. Reversible. Yes, epigenetics is reversible. हाँ या ना? Yes, reversible. Acetylation and deacetylation. Heritable. It is going from generation to generation. Modification of DNA or histone. In this I discussed only histone till now. Without mutation, no mutation. Now modification of DNA. What is modification of DNA? One modification of DNA is, yes, another type. One type of epigenetics is modification of histone. Another type of epigenetics is, yes, modification of DNA. This is not transcription. Okay. Yes. So second epigenetics is modification of DNA. Okay, this is not the answer, because it is not going DNA. This is not DNA replication, this is transcription. Okay. Now modification of DNA, we found that suppose this is a gene, this is promoter area of a particular gene. What I did methylation of DNA at promoter region. If I do methylation of DNA at promoter region, then transcription of that gene is decreased. Yes, transcription of that gene is decreased. Okay. Yes, methylation of DNA at promoter region, transcription of that gene is decreased. So if methylation of DNA, transcription is decreased, you can easily understand transcription. Yes, you know RNA polymerase attached with promoter region. RNA polymerase attached with promoter region. RNA polymerase is attached with promoter region. Okay. This is RNA polymerase attached with promoter region and synthesize messenger RNA. If methylation occur at promoter region, this RNA polymerase will not be able to attach and synthesize protein. Okay, so this is, yes, this is methylation of DNA at promoter region. This gene will not be transcribed. So methylation of DNA, decreased transcription. Question is asked. Yes, in DNA there are ATGC. Yes, so which nucleotide is methylated? We found that cysteine is methylated. Cysteine is methylated. Okay, so sometimes you will find CpG area is methylated at cysteine. Correct? Yes, this question is asked. This is not cysteine, this is cytosine. This is cytosine. So actually this question was asked. Option A they gave cysteine and option D they gave cytosine. So students were so much preoccupied with the word cysteine, yes, cytosine. So you must differentiate this. This is not cysteine, this is cytosine. This is not cysteine, this is cytosine. Good. Okay, very good. Number. So CpG rich region means cytosine phosphate G. Area is methylated. And which nucleotide is methylated? Cytosine is methylated. Yes, transcription of that gene. So this is again another example of epigenetics, that is methylation of DNA. Okay, this is not related to the replication, this is related to the transcription. Again I repeat, at a particular time in a particular cell only few genes are expressed, other genes are kept in inhibited state. Why we are keeping all these genes in inhibited state? When adverse condition would come, we will activate these genes either by demethylation or either by acetylation or acetylation of histone. Then accordingly we will activate the gene, we will synthesize protein will go and will tolerate that adverse condition. That is the concept. Cytosine, this is not cysteine, this is cytosine. So question was asked, they give that methylation of cysteine. Option D, methylation of cytosine. So answer was methylation of cytosine, not cysteine. So many times you are confused. So what is the this is epigenetics. What is the concept of RNA interference? Yes. Now you will understand RNA interference in a better way.